Welcome to Click for IB Biology. We're studying D3.1 and we're now going to look at how the fertilized oocyte develops into the so-called blastocyst and then its implantation into the uterus and the production of the hormone hCG. There's a PDF of this presentation that's linked in to the description down below the video, and there you can download that, annotate with your own notes. But there's a quiz there as well with 10 questions, answers provided, and you can use that to evaluate your own learning. In a simplified sequence of the development, we previously described the formation in which the sperm nuclei and the oocyte nuclei come together, and then they will go through mitosis in order to form and DNA replication, of course, in order to then move to mitosis and the two-cell stage, which takes approximately 1.5 days from fertilization. And by the end of the second day, we have moved to the 4 cells. And remember that by mitosis, this means that each of these cells contains a full set of genetic information. That is, they are diploid with a double set of all the chromosomes of this organism. But after 3 days, we're at the 8-cell stage, and then by this ball of cells here is known as a morula at 4 days, and by 5 days, we have moved to the stage where this morula has started to form a sphere of cells known as the blastocyst, and with a space in here, and this mass of cells here is called the inner cell mass. So we have these membranes here and these membranes here. The fate of which is that the cells in this location here, the membranes rather in this location here, these will form the placental membranes, including the chorion. And this inner cell mass is what will actually go on to form the embryo. And as it progresses to implanting into the wall of the uterus, it begins to secrete a hormone which is known as hCG, which stands for human chorionic gonadotropin. And this is going to be important in maintaining the lining of the uterus, the endometrium. Let's take a look at that in a little more detail. And so here we have our blastocyst here with the potential placental membranes and also the inner cell mass going to form the embryo. And this implants into the wall of the uterus, the endometrium, which of course has been held in this state by the levels of progesterone in the second half of the menstrual cycle, and that progesterone is produced by the corpus luteum. Now, ordinarily at the 28th day of the cycle, this will begin to reduce, but the blastocyst will start to produce the human chorionic gonadotropin hormone, and this sustains the action of the corpus luteum. And so the corpus luteum will continue to produce the hormones estrogen and progesterone, and these will bring about the inhibition of follicle stimulating hormone and luteinizing hormone in the pituitary. And of course, this prevents any more follicles developing as we already have a successful implant. The corpus luteum will sustain its production of progesterone, and that in turn will maintain the lining of the endometrium, which we don't want to break down at the end of the cycle. We want to keep that in place. So again, that is the function of the corpus luteum's sustained production of progesterone because of the hCG. hCG carries on its production from the developing embryo until about the 8th or 10th week, the first trimester of the pregnancy. And this particular hormone can be detected in the bloodstream or the urine and acts as a marker of pregnancy having occurred. And there are reagent strips available. You can see I'm showing the image here of where these reagent strips are showing the double line for the pregnant condition and the single line for non-pregnant. The technology involved in this requires that within this structure here, there are antibodies that I'm going to draw like so. And these antibodies respond to hCG, which I'm going to draw like this. And these antibodies are specific to this particular hormone. When they engage together in the reagent strip, whether from a urine sample, the combination of hCG with the antibody initiates the color change. So this becomes activated and initiates an enzyme, which will then bring about the color change of the reagent here. So it goes from, I guess, from white, in this case, to red. The antibodies are produced by a piece of technology called monoclonal antibodies, which is another biotechnological application of biology. So this is a technique that we can obtain specific antibodies to detect particular proteins, in this case the hCG, giving us the pregnancy testing kit as so. In the next video, we're going to go on and take a look at the placental structure. There's a chance to subscribe to the channel, greatly appreciate it, and there's the playlist for this particular topic, D3.1. Thank you for listening.